Hi, good afternoon. My name is Jude Akwe, and I am studying gustation and sexual dimorphism in the Menuz lab. So, as you may know, many vectors of disease will use chemosensation, that is, sensation through the chemicals in one's environment, to target their hosts, spreading malaria and other such diseases. Studying this process can actually help the development of better repellents in the future. The study of chemosensation can actually help illuminate the host selection process using Drosophila melanogaster as a proxy for hematophagic insects like mosquitoes. One form of chemosensation is called gustation, or taste. There are several sensory organs called sensilla along the wings, the repellent, which is the mouth, and the tarsi, which is the legs of the fruit fly. You may be wondering, why are there taste organs on the legs of a fly? Well, one of the evolutionary benefits of sensilla taste organs, or just sensory organs, being on the tarsi is that it allows the fly to actually taste its food before ingesting it, so that it can prevent the ingestion of potentially hazardous chemicals, usually indicated by bitter compounds. So, it's preventing itself from, it can taste poison before actually eating the poison. Um, So here is a picture of what that would look like. And here is what it looks like for the sensilla to actually be aligned along the leg. And then here is a more blown out image of what a sensilla looks like. So the tips of sensilla are either straight or forked in morphology. Depicted here is a straight tip sensilla. And they have a central pore through which the tastins or the ligands will actually enter through. Once the tastins or the ligands have entered the sensilla, they are received by the gustatory receptor neurons depicted in the pink and purple, and that sensory input is then directly sent to the brain in the case of gustation. Surrounding the gustatory receptor neurons, the GRNs, are the support cells shown in the green and light green. So when it comes to gustation, there are several different types of sensilla. But of chief importance, though, are the gustatory receptors and the ionotropic receptors. The main difference between the two are what kind of ligands actually activate them. But although we know what types of receptors there are, many of these receptors are actually termed orphan receptors, meaning that it's unclear what specific ligands, what types of tastes, what specific molecules actually activate them. Likewise, there can also be many orphan ligands and even orphan gustatory receptor neurons. So, as you may already know, taste and other such senses are extremely vital to fly behaviors, especially when it comes to courtship. In Figure 2 is an example of that. We see that typically males will taste females for any pheromones to detect if she has yet to mate or not. This difference in behavior during courtship is an example of sexual dimorphism. So, if we think back to those orphan receptors, one could reasonably ask if we'd be able to use sexual dimorphism to de-orphanize these receptors. In other words, identify novel pheromone taste receptors in the fly genome. How might we be able to identify said hypothetical receptors? We can do this through differential gene analysis, right? So we're thinking back to that sexual dimorphism. We're going to collect RNA sequencing data of male versus female flies for their gene expression of those aforementioned orphan genes. Thus is the goal of my current independent research project. So, to conduct this study, the following workflow is implemented. First, we're doing a lot of dissections. So Figure 3 is a schematic of how those dissections actually worked. We're dissecting 4 samples of the forelegs, mid-legs, and hind legs. Per sex. So for the end, there should be 24 samples in total, and each sample should contain 200 legs. So then we've extracted the RNA for all of those tissues, for all of those samples, making sure that there's no genomic DNA contamination. And as a check, we can convert that RNA to complementary DNA just to make sure that there's no genomic DNA contamination. And we can also send that complementary DNA through quantitative real-time PCR just to make sure that we've collected the tissue that we actually need and not fly head tissue. So, after all the RNA extractions are completed, we will send that RNA to UConn CGI for RNA sequencing. And then in about 2 to 3 months, we'll get that data sent back, and then we can finally conduct that differential gene analysis. However, during the summer, we've encountered some problems with the RNA extraction protocol. So we had to do a lot of RNA extraction optimization. Figure 4 is showing those kind of problems that we saw. So in order to not waste any of that vital leg tissue that we need for the actual study, we practiced with extracting the RNA of fly heads. So when you're extracting the RNA of fly heads, you want to see an overall RNA concentration of about 200 nanograms per microliter. But as you can see with figure 4a, we're seeing concentrations of about 15 to 16. Additionally, we saw really high salt protein contamination and really high salt contamination. That was with the original QIAGEN kit and QIAGEN protocol and grinding the tissue with plastic pestle. So in figure B, we show how we try to remedy that problem with using a Downs homogenizer instead of a plastic pestle to more regularly grind up that tissue. And we found that although the overall concentration of RNA did increase from about 15 to 16 to about 74, as you can see, still a lot of protein contamination, still a lot of salt contamination. In fact, with the orange curve, you can actually see the actual salt that's in there called guaiodinine, and that is the actual name of the chiotropic salt that's used to prevent the RNA degradation of your sample. So that just goes to show that the salts aren't really eluting out of the samples. So then we try to throw away the entire kit and replace it with the purine kit in the purine protocol. And we found that all the problems were solved. The RNA concentration increased to about 200 or 160. No salt contamination, no protein contamination. So then we tried to find out what was the problem, right? So we used the new purine kit and we used one of the QIAGEN homogenizers, and we found that although the concentration did increase a little bit, there was still, as you can see, a lot of salt contamination, a lot of protein contamination. So we can reasonably conclude that it was probably just due to the old QIAGEN kit being a little old. So now that we have all of that sorted out with the RNA extraction process, we can now finally finish extracting all that RNA and send it to UConn CGI for sequencing. And then we can finally actually hopefully identify some of those genes that are differentially expressed between the sexes. So I want to thank Dr. Menuz and the Menuz lab, especially Prayaji Mohapatra, who was the grad student that I studied under for this project, the MARC program and NIH for funding this study, as well as the McNair program and LSAMP. Thank you very much for listening.